8:30, and that's where we always meet here and to celebrate vision research. And today we have a very, very special guest, Anand Swaroop, who is here coming from another coast, and we couldn't be more delighted to have you here and to show you what we do, and also to have you in person deliver your thoughts about what is important and what is not. So with that, I would like to pass to Sam to introduce you for one.
Good morning, everyone. It's my distinct pleasure today to introduce the preeminent scientist, Dr. Anand Swaroop. Today's distinguished speaker who will be speaking today on the nexus of genes, aging, and environment to determine disease phenotypes with specific lessons from age-related macular degeneration. Dr. Swaroop is the senior investigator and chief of the intramural neurobiology, neurodegeneration, and repair laboratory at the National Eye Institute in Bethesda, Maryland. Received his BSc in chemistry, zoology, and botany from the University in 1975, his MSc in biochemistry from GD Parent University in 1977, and his PhD in biochemistry from the Indian Institute of Science in 1982. He then moved to the United States to pursue postdoctoral training in the lab of Alan Guerin at Yale University. He underwent subsequent training in the labs of Dr. Uda Frank and Dr. Sherman Weissman before starting as an assistant professor in the Department of Ophthalmology at the University of Michigan in Ann Arbor. He rose to the ranks of the University of Michigan, where he served as a faculty member in numerous programs and centers before being recruited to NEI as a senior investigator and chief of NNI in 2007, where he has been ever since. Of course, he has been heavily involved in service to the community, including by service on study sections, giving grants for foundations both in the United States and abroad, and serving on the editorial boards of numerous journals. He's also won numerous awards for both his research activities, such as numerous NEI Directors Awards and the Alcon Research Institute Award, and his advocacy activities, including numerous diversity and mentoring awards from NIH and NEI. He has filed numerous granted patent applications and disclosures, and has even successfully competed for grant support while at NEI. He has a current Google Scholar H index of 95, has published over 390 papers, and has been cited over 37,000 times, indicating the depth and breadth of his continued contributions to the field of vision research. His major interests and contributions to the field include genetic and epigenetic regulation of retinal development, aging, and evolution, IRD models and therapies, AMD, and stem cell modeling and therapies for retinal diseases. Please join me in welcoming Dr. Anand Swaroop to our seminar.
Meeting with Lefkowitz, and somebody cited like, oh, he published 600,000-700,000 papers, whatever number, and you have a large number. Did you read all of your papers?
That's a different question. We'll discuss that over dinner one day. First of all, let me thank you, my brother, and I think more than you, I'd like to thank Greshna, who actually invited me, and so shout out to her. And also to Anna, who has been taking wonderful care. She's right there. So thank you very much. It has been really a pleasure. And Chris showed me around and the beautiful center that and the research program that he, Greshna, and others have developed and recruited a bunch of wonderful scientists recently. So I couldn't be more proud and happy. And so thank you very much for inviting me. And I'm honored and delighted with the wonderful introduction and everything else. So what I'm going to do today is share with you some of the work that is going on in my lab. We do many different things, but this is really an exciting focus during the last few years. What I will do is I will sort of give you a little bit of historical account as well, and then move on to show you some data. Reintroduce you to some of the basic concepts because this could be a diverse audience, then show you some unpublished data, and then end with some thoughts, some speculations, and I hope you'll allow me to indulge in some of those. So you can see there'll be a little bit of an overview, but I'll be happy to discuss with you and others or during discussion. So let's just sort of get started. Let me see. Yes. I always like to show this as the first slide in many of my talks, particularly related to genetics. And if you see on the left here, that shows the uniqueness of all of us. In this room, every one of us have very unique characteristics. All of us have a lot of similarities. We have nose and ear and hair and sort of similar looking body structure, but still we have different shapes and sizes, and we come in all different ways. How does that happen? The information to create us is really within the 3 billion nucleotides, but probably about only 20,000 or so genes, give and take a few thousand. And many of these are even in yeast and E. coli and wherever. So what does make us unique? So I've been sort of very fascinated by some of those questions, that how these 20,000 or so genes create a unique individual that we are. So a few years ago, I was very fortunate to be part of this 800 author paper in Nature that was from 1000 Genome Consortium. And this particular paper looked at the variations in our genomes. And these were from 26 different populations at that time by using whole genome sequence. Since then, this data has been really refined, and we have a lot more information. But what it tells you is that our genome is not a static entity. All of us also have numerous variants, numerous different changes. Actually, some of us, or actually all of us, probably have about 10,000 variations from whatever is called as a reference genome. And 10,000 of these variations are unique to us. Maybe our parents and brothers, sisters share that, but nobody else has those 10,000 or so unique variations that we have. Majority of these variations, of these 10 million, we think are benign. We think today, who knows tomorrow what will happen. A few years ago, we used to talk about nonsense genome or selfish genome or whatever, whatnot. Now we know that the non-coding regions of genome are probably even more important. So these variants, many of these variations give you the uniqueness, unique identity that you have, but many others can cause disease phenotypes, can cause problems in certain, they can perturb certain cellular reactions. So how does all of this happen? So let's sort of think about it. What is disease? So if you want to think about disease, we need to first think what is normal. Before you understand what is normal, or rather what is abnormal, you need to know what is normal. If you look at on the right side, you have this genome, 3 billion nucleotides packed in 5 micron or whatever different size nuclei, very tiny little sort of compact structure. And somehow it opens up in different cells, it turns on distinct sets of genes, and that transcriptome or those transcripts, many of them can function on their own, as we know today, but many others generate proteins. And these are this huge network of these transcripts and proteins and other entities that you have. And these complexes, they together form this very intricate, complicated network. And that network is very tightly controlled. If you have one variation or change in some specific cases, that can perturb lots of nodes around that. So it's not that none of the genes or proteins work alone in the cell. They're all connected to something else. So until a few years ago, in my own lifetime, some 20, 30 years ago, we used to think of working with one gene or one protein. And we used to think, we solve it. And we did. A lot of us did a lot of work, and we found some very interesting sort of functions and all. The trouble is those functions don't exist sort of as a lone entity within the cell. They form these very complicated protein complexes or RNA protein complexes and these networks, and they carry out these reactions. So what you need to do is you need to do a very systems-based approach. You need to look at the whole thing together in order to understand the organismic behavior, that is human behavior, the uniqueness of human identity. And in our case, that identity really belongs to retina, right? That's our focus. So what we are going to do is we are going to think about that. But in general, the disease happens when that normal, very tightly controlled network is disrupted, perturbed in some manner, right? So if we want to know that, we need to map all of those things. We need to understand it completely. We have to have a very holistic view. So let's talk. This is the only clinical slide I'm going to show because I'm not a clinician, and I have wonderful, wonderful colleague down there, Emily Chu, who teaches me all the time, you know, what macular degeneration is. So all I'm going to show you on the left side, some fungi from individuals from macular degeneration. Generally, all this looks very beautiful, no perturbations, nothing. But even I, not a clinician, can see something is wrong here. There's some blood around here, some atrophy around there. Something is going on. So there are huge perturbations that are happening. And what that does is that those perturbations are leading to loss of central vision in case of age-related macular degeneration. But this could be true for any disease. It could be actually true for even single gene Mendelian disease or a retinopathy, where even though we believe a gene is causing the disease, the phenotypes, even with same mutations, can be very, very different. Okay, so whatever you want to call it, the bottom line in these cases is that this is very complex kind of phenotype, and the severity and age of onset can be different in different individuals. There's extensive variability. And in advanced stages where you get blindness or loss of vision, we call it geographic atrophy or choroidal, or there's some neovascularization and names are changing. And again, I don't want to get into that. All I want to get into is the basic fundamental, how does that happen? If you think about it, we know that the progressive disease, you can have anything, but you don't get it when you are a child. AMD is age related, so you have to be old. Okay, so it's progressive. It's also very complex. It's not a single gene disease. And a lot of times, scientists, all of us, including me, are very reductionist. We try to sort of distill everything into some very simple way to understand that. And there's no problem with it. Unfortunately, it's not as simple. And it's multifactorial. And I'm going to explain what that really, when I'm saying that, what it means really. So multifactorial means there are multiple components, multiple factors that can cause the disease. And in this triangle or trifecta, what I'm showing you is on top actually aging. Because to me, it appears that advanced age is probably the single most important factor that leads to age-related macular degeneration. Along with that, and until a few years ago in my own lifetime, when I started, nobody believed that AMD is a genetic, there is any genetic component. Only when a bunch of wonderful studies from many people, Jonas Seddon and many, many others, showed that by twin and familial studies that there is a genetic component, and Mike Klein and others begin to collect families and say, yes, there must be something going on there. So there's a susceptibility. It's not a single gene, but there are multiple genes. And that too, variations in those genes make you susceptible to disease. So these are not AMD genes, they are those AMD susceptibility variants. And then there's a major impact of environment, which is sort of, we lump there almost everything that we don't know, right? You get smoking, diet, lifestyle, exercise, this, that, anything that you don't know, how we change our behavior, where do we live, socioeconomic status, could be anything. All of that actually impact macular degeneration, onset, severity, and all that. So let's just first talk about genetics. And since a lot of that is already known. I'm going to just run through some of the history because some of you students here need to appreciate how complicated that has been. I told you already, people didn't believe there's genetics involved in that, but a lot of people begin to do a lot of collect cases and controls and families, and with a lot of work, things were going slowly until these pioneering papers in Science came, and there was an association of a variant Y402H, famous Y402H in complement factor H. And these landmark papers and subsequent papers showed that this is very strongly associated with macular degeneration. Again, remember, associated. Nobody said it causes the disease. It does not cause the disease. Remember, AMD is multifactorial. You can have all the variants, may never get the disease. And that was based on some initial very small jivas. And then there were larger jiva studies that were done, Johanna Saren's lab and my lab. We published 2 big papers using few thousand individuals. Not too many, 2,000, 3,000 kind of individuals. And we added a few more loci. And along the way, Rando Elikmats and many others used candidate gene approach and added a few more complement genes to it. So now we have maybe 8 or 10 or some genes of that time, but we realize that we are getting stuck here. We don't have enough power. So many people join hands all over the world. And this, I highlighted that there were 13, I think if I counted correctly, or so groups that shared whatever jivas data they had. And there was a jivas meta-analysis that was done. And that jivas meta-analysis, this is Manhattan plot. It shows those tall buildings like in Manhattan you have. And so above a certain sort of threshold, you say that is a significant association. And based on that, there were 19 or 18 or 19, I think 18 have been replicated associations that were shown at that time. And this particular jivas meta-analysis through this consortium added 7 more. Great. You have a few more. And then we realized that this was a meta-analysis. It was insufficient. NEI paid extra funds for a collaborative venture by this genetics consortium. And there were a new chip created at Hopkins that there were 16,000, 17,000 controls and 16, 17,000 sort of AMD individuals, advanced AMD individuals, mostly advanced, and the DNA from that was used to do this chip analysis, which had rare variants and some rare variants, but mostly common variants. Jivas or genome analysis studies are generally done with common variants because they give you more power. And based on that, this was the major paper that came out in 2016, and that still remains. Few more genes have been added, few more loci have been added by a bunch of people, but this is the latest, very replicated study. And now you have 34 loci. When I say locus or loci, that's a region of the genome. It's not a variant. You have a lead variant, you have a main variant, which shows the highest association within that region. But then that region, that locus, along with that lead variant, has other variants in what we call linkage disequilibrium. Okay, so even though you have one lead variant, but if there are another 100 variants in LD or linkage disequilibrium, actually any one of those variants could be associated or could be contributing to disease. It's not just the lead variant. Lead variant is more likely, but these are all statistical associations, remember. So here in 34 loci, you now have 52 variants, independent variants. And some loci, you have more than one. The trouble is, we still didn't solve the problem. And my friends, clinical colleagues, and others begin to focus on one gene or two genes, CFH and ARMS2, and say, oh, you know, if you have this, what happens? How it changes the phenotype? The problem is AMD is not just CFH or ARMS2 disease. It's just, we just showed there 34 loci, 52 variants, and maybe many, many more actually. from new studies. So how does it help anyone? What do you do with this? So the question is, what does all this mean really? So we just have to first ask, take a step back and we ask, what is the question? Why this is so complex? So one problem is that GWAS is done with common variants. A lot of us inherit many of those variants, but we never get disease. It's just used for genetic association. So it's a statistical association. And a vast majority of lead variants, in fact, are in non-coding regions. They're not coding regions. If they're in coding regions, you say, it changes this amino acid, this changes this, whatever. But no, they are non-coding regions. And actually, even for CFH, we published that. Immediately after CFH papers came out, we showed that CFH haplotype without Y42H actually show even stronger association to susceptibility of AMD. So there are many non-coding variants in CFH regions which are associated with disease. And then I told you with variants in LD. So there's a very unclear causal relationship. Whatever we want to believe, we can believe, but the fact is that we don't even know what is the relevance of many of these until we show. We need to do something about it. So just sort of, again, logistically, you just make a hypothesis. As scientists, we try to make a hypothesis and we say, what is AMD? We say different variable phenotypes in AMD are caused by cumulative and contextual, context-dependent impact of these variants in relevant tissues or cell types and on specific pathways. Now, there's a lot of things, RPE is clearly probably the most important cell type, but that's not the only cell type. We don't even know whether the disease begins in RPE. It can begin anywhere, but it shows its phenotype. The problem shows up in RPE first. It could be in endothelial cell, corollensis, could be in photoreceptors, could be in Muller glia. I don't know. Nobody knows because we don't know how AMD begins. We know towards the end based on pathology, based on huge amount of wonderful studies that have been done by amazing number of investigators. In genetics, each of these variants that we have individually likely has a small effect on disease, on gene expression, because they're non-coding genome, and even on the function of specific target proteins. So we speculated, we sort of put together a threshold hypothesis, multi-hit, few of us, and in this review in 2014. And some of these just serve as a framework. What we said is that maybe variants in different genes, they are associated with different pathways and function in different cell types, and eventually lead to variable phenotypes in a cumulative way. So we sort of put together, we said you need multiple hits. Maybe you need to hit 3, 4, 5 different pathways in order to get the disease, just one. And we showed in that there was a genetic analysis done where you can have all complement pathway variants, but still you may not get disease. There were individuals who had all high susceptible variants, but still will not get disease. So, and there another is this, ARMS2 haplotype. I mean, I am homozygous for ARMS2 haplotype. Being a scientist, I had my whole genome sequenced as well. So I'm ARMS2. And so, that's very serious. My father had AMD, so I'm susceptible definitely, but something is still missing because I'm 65. So far, so good. Emily is going to check me out. I think later this month, once again, I go to her to test that. But the fact of the matter is, you need so many different things. So one possible solution from genetic perspective is that, if you can find rare variants, like in retinopathy, single gene disease, you find very rare variant, which have very high genetic burden, we might be able to say, this is the gene, this is important, this is, in this area. Just having a lead variant next to a gene doesn't mean that is the gene. It could be 1000 kb away. So if you find rare variants, so that needs large families, you can do whole exome or whole genome sequencing. And if you have it in the coding region, it will be easier because you can say, this residue is conserved and this is causing. So we did that thanks to my client and also to Emily Chu and Lena Zellinger, who was a fellow in the lab. She's still working as a special volunteer. So basically, Mike has got over 150, I mean, I don't know, the hundreds of families that he has collected, small, big. This is one of the large families that he had. And we have done whole exome sequencing for 106 and whole genome sequencing of 20. And the analysis, as you can imagine, is very complicated in this case. And even if you find all these variants in coding region, what does that mean? So we had to put together certain threshold. We need to validate. And one of the first paper on that, we have just submitted, I think it should be coming out. There's another complement gene involved in that. So, and there are more, hopefully it will help us understand the value of different genes that might be there, but still it doesn't tell the full story. So let's come back to AMD associations. As I said, a vast majority are in non-coding regions, and there are variants in linkage disequilibrium. How can they impact disease? Simple idea is that somehow they have effect on gene expression in a quantitative manner, right? That's what, or could be qualitative too. So let's go and understand basics of gene regulation. This particular cartoon shows you complexity of just transcription. So you have RNA polymerase II, which binds to promoter regions near the initiation site. Somehow it travels, but then stops. There's a pause, but until other transcription factors and regulatory proteins come around, then it can move to elongate the transcription. And then it changes its behavior. Now, Pol2 has got phosphorylation. There are tons of signaling proteins involved in that. And when RNA is being made, there's splicing factors that come into play, and there's a coupling of transcription with splicing, which helps with elongation as well. And then you have the termination. Much of the work has been really focused on initiation, but there are many, many other steps that we don't even understand. And you can have problem at any step. Think about it, variant at any of these places in some way can complicate the story. Let's talk just initiation. And this is a very old cartoon from Walter Schaffner, who discovered this enhancer, who coined this term enhancer. So these enhancers can be direction independent. These enhancer elements can lie hundreds or even 1000 kb away. Some of these interactions or regulatory elements could be in trans as well, could be on different chromosomes even. But let's talk about cis element. Then you have insulator boundary element. And if you have variants, just think about it. If you have variants in any of these cases, they can somehow impact transcription of the gene that you're looking at, that you're interested in. So the gene next door does not have to be the main gene. And then how do those things come together? This is looping, and you can say this, as I told you, 3 billion nucleotide long DNA compacted in a small nuclear structure. And what happens is some of these enhancers form these loops to bring those regulatory elements close to the promoter. And they form very sort of high concentration, high density protein liquid condensates. And these condensates, there is a promoter condensate, where it moves, it changes its composition, has different sets of proteins. And now you have another condensate, the elongation condensate. And then splicing factors come into play. There's just tons of this complexity there. So now think about variant. A variant is far away, like that, those variants in those with the stars looking there, they can look at they can control the expression of a gene way down. And if that can be done in a quantitative manner, this is called EQTR. expression, quantitative trait, locus. So what we did was, we did not just focus on AMD, we took the whole retina and then we did the EQTL transcriptome analysis, we did EQTL analysis. This was published a couple of years ago in Nature Genetics. And thanks to my friend Deb Farrington, who had meticulously collected literally hundreds of retina samples. So she gave us punches. This was just very interesting story, just walking around in a genetics meeting. I was grumbling. I said, it's so hard to get human retina samples. And Deb and I were chatting and she said, how many do you need? I just sort of thought, oh, maybe I said, 100, 200 will be sufficient. I just added some more. I was looking for 30, 40, but I said, maybe 100, 200. Yeah, okay, let me go back and check. And she went in her freezer, said, I think I have close to 800 or 1,000. How many can you send? No, I can't do that many. So we took 500 post-mortem retina punches. She also had macula and RPE from many of these, and she sent us those, but we focused first on neural retina, peripheral retina, and we did genotyping of all of these. We did transcriptome analysis of all of these, and working with a couple of very wonderful bioinformatics and statistical genetics, Nilanjan, particularly Chatterjee at Hopkins, we showed that there are 10,474 genes under genetic regulation. So there are variants far away that can control the expression of these genes. And many of these, over 4,000, 4,500 or so, are retina-specific EQTLs. They were not in GTEx. They were not in the database which studies all other tissues and all. So this is published. So the bottom line is now we say, now you can combine the GWAS data from AMD on this and see how many of these could be important for AMD. But this data can be used for other retinal diseases as well. Okay, it can be retinopathy, it could be anything. But this still did not tell the story. There were variants, and with Eilert Segre at Mass Eye and Ear, she took that data and began to look for variants which can affect splicing. Variant doesn't have to only control expression. So what she has found by all of these, the huge amount of study, and in peripheral retina, 2850 S genes that are controlled at the level of splicing by variant. And we have done the macula. I told you we have macula. That data we have not fully analyzed, but she has done for splicing the 3090 macula. So, and there are differences in peripheral retina and macula SQTLs as well. So let's come back, because we didn't tell the whole story yet. Okay, I don't know, I don't have, okay, so we start. So let's come back to this original thing. So even though you have EQTLs, they are far away, how do you know that they are functional EQTLs? How do you know they really, yes, you know that EQTL, that variant, at that point, when we did the expression analysis, maybe correlated, is statistically associated to expression. Can we show that that variant really indeed is connected to the promoter of that gene, right? That's a little bit more functionality. So again, coming back to that 2 meters of DNA in 3 dimensions within a tiny, tiny nucleus, yet it functions. It creates knowledge, it creates information, it transcribes specific genes in a very stringently controlled manner in every cell type. And anytime you have a problem, you get disease. So how are these genes organized? I told you about loops. There are enhancer promoter looping. That is a lower level organization, or you can say highest level organization. Some of these loops, enhancer loops, can also have many enhancers, some of these looping, and you can have super enhancers where many proteins, many enhancers can come together. And then there's another level of organization, what we call topologically associated domains, or TAD. And when many of these loops are sort of part of sort of certain structure, a domain, and that domain is sort of having some sort of boundary element. They are made with cohesin and other sequences where the gene expression generally is restricted within these boundary elements, and so those are topologically associated domains. And then you have larger compartments. Until recently, we all thought that compartments are very large. They'll be in megabase structure. You all know about euchromatin and heterochromatin, right? So heterochromatin is sort of inactive sort of region of chromosomes, and euchromatin is active, right? So, but we call it compartments. So these different topologically associated domains are forming together probably these compartments, and these compartments could be megabases long, and compartment A is generally euchromatin and B is inactive. But it's not as simple. There's a paper in bioRxiv which has done very deep analysis. Most of these is based on Hi-C analysis. Hi-C is the genome-wide analysis of chromatin conformation. And when you do that, depends how deep you go, you can do the resolution. Most resolutions are 50 kb long, but 50 kb in a genomic region could include so many different things together. So this particular group did 0.5 kb resolution. For that, they had to sequence, I don't remember, some 10, 15 billion nucleotides or something like that. And with 0.5 kb resolution, what they were saying is that even within the same gene, you can have different, the gene nuclear, the DNA can go into different compartments. Some parts could be in B compartment, some could be in A compartment. And theoretically, this sounds so fascinating. Many genes have large introns. Why should RNA pol 2 really transcribe each and everything, right? So what easily you could do is put part of your intronic sequence in B compartment so that RNA pol 2 can sort of move on and to go to a different region. I mean, it makes sense. We don't know that yet because that's only one paper is 0.5 kb, everything else is much higher. We did that. We thought we'll do that for retina. Most of these studies were done with cell lines and cancer, this, that. Very little is available with tissue, if any. So we did that with retina. And what we did was we did Hi-C and we did deeper sequencing. We did high resolution of 3 kb, which is pretty good, not so bad. Means if things are out more than 3 kb apart, then we can distinguish them. Within 3 kb, we cannot. And those that's the way you look at. I'll show you the next slide. Basically, this high resolution data tells you different chromatin contexts. And we did 1.15 billion base pair sequencing. And we also identified about 3,000 TATs by this analysis. And then looking at the loops, we have more than 60,000 loops. But then we took data, some of that we created, and some of that came from Tim Cherry's beautiful paper in PNAS. And we looked at chromatin states, and we did also analysis of super enhancers. We looked at sequences, super enhancer sequences, and then we combined all of these data. We wanted to see which super enhancer is connected to what gene or what sequence at a 3 kb resolution. I mean, I wish we could do more, but we'll have to go to 10 billion to go further. So we did integrated mapping of all of this, and then we took AMD GWAS data, and then we also incorporate actually glaucoma GWAS data, but you can do myopia, you can do, and you can actually put retinopathy genes. Basically, this data now can allow you to look at regulatory element for any gene that could be very far away, that you have, generally you look promoter analysis and enhancer very close by 2 kb, 1 kb, you do, but that even if they give you specificity in your transgenic mice or whatever, it's not the whole story, because you have to look at in a more holistic way. In any case, I don't do any of these things, so I have to give credit to people who do. And Claire, Zach, also Jayashree, these three are top-notch bioinformatics, statistical genetics people. Nivedita did the Hi-C with Jimena, and Kathy did the ATAC-6. So with all of these wonderful people, what we did was it just came out last month. So we basically have, after merging different high-C data sets from different retina samples, 4 retina samples, even though we have 3 kb resolution, we called the loops at 5 kb because we were a little uncertain about some of these. These are not uniform. You have to understand, in the genome, these things are not distributed equally, like, you know, every loop or TAD is sort of nicely. There are areas where you can have very high resolution, even 1 kb, and then there are areas where you have resolution that is 10, 20 kb. I mean, you know, that's just the nature of the beast here. So the chromatin interaction, then we compared that to mouse high-C data from Mike Dyer and from others, and basically said there are a lot of conservation you can look at. So this is how you look at the high-C data. This is a low resolution, next level of resolution, and then you can, each of these dark spots that you see here, the darker the spot, higher the interaction. More clustering means high interaction. So that's really what you have, and you look at every gene, and these things indicate the loops. These things. So enhancer could be here, but it can, if 1 foot could be in one place, some 1000 kb away, but it could regulate the expression of a gene way downstream. So I just want to show you, this is not pertinent to this particular talk, but this just shows you that super enhancers are rich in chromatin looping, and they have tons of active chromatin marks. So, you know, active chromatin marks are K27 acetylation and K4 trimethylation kind of marks that are generally seen. But also, thanks to Tim Cherry's data and some of our data, by combining all that, you can look at, and this is a sort of very zoom out view, right? It's very condensed. Now you can just zoom in the data, and all this data is available, basically publicly available. So anybody can look at any gene, and if you want our help, you know, we'll try to help you, show you something. But basically, any gene you can look. You want to see rhodopsin, where the enhancers could be. It could be 1000 kb away. You never noticed it. So in a way, that's very important. Just think about it. There's a missing heritability. Not every individual have got both mutations. In some cases, the homozygosity or a compound heterozygote. You know, the other mutations in some of those individuals could be in the enhancer region or super enhancer regions and all, right? So theoretically, not theoretically, you can really take every region on the genome and do the sequence analysis, and we are trying to do that. It's sort of a lot more complex than I said, but it takes time and effort to do it. But you can take those and you can look at all the individuals that have already been sequenced and see if some of those have variants in those enhancers, and then you can analyze whether those variants can impact the expression of the downstream gene. So now this is sort of just to put all of these things together. You can have a variant far away. It can be linked to a gene through a cis regulatory element, or it could be through a loop, chromatin loop, very far away. And any variant that can affect this looping, or that can affect some of these interactions or proteins that bind to these enhancer elements or whatever, can actually reduce or increase the expression of a downstream gene. So this was done with AMD loci, as I said. Just think about this particular locus. Here is one locus. This is the lead variant, okay, on chromosome 20, I think. And, you know, it was said which gene is there, and I have to wear my glasses, presbyopia, but, you know, it was said this is C20 or 85 locus. But we don't know anything else about it. So if you think about it, there are many other variants around this. These are the variants in linkage disequilibrium. And look at this. There are tons of these things. They're looping from here in this region over there. If you can zoom in, these are the targets likely of that variant. not C20 off. C20 off can also be because it's right there nearby, but probably not because it's a little far away still. It's the closest gene, but more likely are these genes. And you can test them now, whether they have something to do with it. And some of these can, so again, as I said, right now, this is on an average 3 to 5 kb resolution, but in some areas it could be very, very high. In some other areas, it could be not so high. This is for glaucoma. We added glaucoma loci in there too. And in this paper, we added some examples. But you can look at any gene. It doesn't matter. But you can put the variant. You have variants in linkage disequilibrium. And then you can start sort of saying where those variants make contacts and where those contacts other end foot is and what genes might be there. And you can then test this hypothesis. All right, this is one part. This is genetics. We talked about genetics. Now, more complications. This still does not tell you the story. Let's talk about aging and environment. Aging is very complex also. Aging is multifactorial. Not everybody ages in the same way. There's genetics to aging. There's also environment to aging. People age differently, living in socioeconomic status, geography, where you are, what you eat, right? It changes. So all of these, at least in part, and there have been a lot of studies that show that at least in part, some of the impact of aging and environment is mediated through epigenome. And the epigenome that is a little bit more stable and more accessible for study and easier to study is DNA methylation. So people have studied DNA methylation with respect to aging, with respect to environmental sort of disruptions as well. Environmental Health Institute has got huge amount of funding on that, and same thing is true with Aging Institute. So there are a lot of epigenome. When we talk about epigenome in this case, primarily DNA methylation, because histone modifications and other epitranscriptome can change much more rapidly probably. And this is a little bit with age, it does change. And actually we did show that. This is Jimena Corso-Diaz in my lab a few years ago did very nice study using purified rod photoreceptors from young and old retina. And those retina, when she did the comparison, she found very specific gains and losses of 5-methylcytosine. And this is just a sort of Sarko's plot of this age-related DMR or differentially methylated region density. And you can see there is some clusters. There's a cluster here on chromosome 10, and that's where the longevity genes sort of are there. Many of these clusters were in the mitochondrial gene. I was talking to Chris yesterday, and I was saying, we are coming here in very different angles. A lot of metabolic genes, a lot of mitochondrial genes over here. There are a lot of the nuclear coded mitochondrial genes. So there's something happening here with aging that affects. So we thought we can do that for human because we thought DNA methylome analysis can allow us to link genetics to aging and environment, right? And there have been papers, genetic variation influencing DNA methylation, providing certain molecular mechanisms and all. So they have done in different tissues. It has to be actually cell type dependent, but sort of we don't have that kind of capability yet at a high throughput level. It will come maybe in a couple of years, but right now we could do that at the level of retina. Just two terminologies, MEQTL is methylation QTL. Actually, initially people were using MQTL, but M is now being used for metabolic QTLs as well. You can use, what that means is you have a variant that affects methylation. And in human genome, a lot of methylation is in the CPG regions as well, which are scattered and depending on, it's a different story, but I'm not going to go there. But the bottom line is you have variants that can affect CPG methylation. And then you can have variants in CPG methylations that can affect gene regulation. They are called EQTMs, or expression quantitative trait methylation. Okay, so remember these two terminologies. MEQTL, So you saw EQTLs, SQTLs, now MEQTLs and EQTMs. So here is again Deb Farrington, my friend. She had all these retina samples, retina and also macula. So we have done that. And this was done with Arlette Segre again, who is our wonderful collaborator from Mass Eye and Ear. And she actually is the one who's guiding Jayashri. I am incompetent to, I can understand probably and maybe interpret a little bit, but the analysis is all done by Jayashri and Arlette. And what they've done is they've found MEQTLs 37,000 plus. That means variants which affect methylation at these sites in the retina. Some of them are similar to others, some of these are not, some of these are retina specific. And this is just a distribution where they are. MEQTLs are generally, if you look at that, they're in the spliced donor areas, they're in open chromatin region. Okay, again, gene regulation, right? They are in the frame shift regions, they are in the region of CTCF binding. So you can see where they are not in some of these other areas that you would think. They're just very clustered, these MEQTLs, because they are also related to CPGs. And one of these genes that we are showing here that is coming out is PARC7. So the MEQTL in the retina that affects probably methylation at PARC7 gene, which is very important gene. Now EQTMs are even more interesting because these are the CPGs that can affect gene expression. And there's a non-random distribution. That means these EQTMs are not scattered all over the genome just like that. They are really, really clustered in certain region. And they found Jayashri, they means Jayashri and Arlette, showed that there are 13,000 retina EQTMs. And you can see the correlation of these EQTM. This is with NLRP2 inflammasome, one of those. This is with GST-T2B, this is glutathione transferase gene, one of those. So these CPGs, changes, variants can affect gene expression, and that shows the correlation either up or down. And this is a distribution of EQTMs on different chromosomes. This is non-randomly distributed. Chromosome 1 is very big, so yeah, there are more. But look at these chromosomes, 16 and 19 and all. They are smaller chromosomes. They have a lot of EQTM. Then we also looked at EQTMs, some variant that can affect many different genes, right? So methylation doesn't have to, it's not like one CPG methylation variant can affect only one gene. It can affect maybe 10 genes, it can affect 100 genes, I don't know, it can affect many genes. So you look at the distribution and the number of CPGs with more than 10 EQTMs were really highly present on 16 and 19. And if you look at that, and if you look at the genes in these regions, where these clusters are, and if you can look at the genes, look at the pie chart. What does it show? What kind of genes are there? I can't read. I have, but I think there are mostly genes which are transcription regulation related, metabolic genes, mitochondrial genes. I'll have to wear my glasses to look at what they are, but you can read it much better than, yeah. Look at the metabolic genes and transcription genes that are very highly sort of regulated by some of these methylation, CPG methylation changes. So if you have a variant, just think how beautifully nature has designed. All you have to do with age or environment is change one methylation. And that one methylation can change 10 metabolic genes. Beautiful, right? Rather than just sort of, having 10 different changes and all. No, just one change and you can do all that. So, I mean, that's really, I'm fascinated by some of this data that is coming out, and now one can go in and one can look at what's going on. There's a lot more complexities. This is all unpublished data. This is all very new. And I'm also beginning to understand it sort of slowly and argue with Arlette and Jayashri and others to figure out, what it means. But we are interested in AMD. You can or glaucoma or whatever you want to do with this data. So we said, okay, let's do a combined analysis. Okay, this is called summary database Mendelian randomization, or SMR. What you do by this is you can look at GWAS of AMD with MEQTL, GWAS of AMD with EQTL, which is what we did earlier. And then you can also look at EQTL, MQTL relationship and MEQ, so basically you can do this kind of correlation, but SMR can do only 2 correlations at one time. When you do some of these correlations, what you find are this kind of analysis, this kind of stuff that come, and the genes that are showing up are associated with glutathione transferase. Now, why I picked up this example? A lot of biologists, a lot of people who work on different systems will tell you genetics has never found any oxidative stress genes. Genetics have never found any stress response genes, and you find all the other stuff. But my data shows oxidative stress is important. Jim Handa will tell you smoking affects all these genes. But where is EMT? Where is this? Where is that? That's where it is. That doesn't have to be genetics. That's environment. So if you do the MEQTL analysis, now we are beginning to find genes that we did not find in genetics. We can also do multiple trait co-localization or MOLOC analysis. And I pulled out the slide because it'll take a lot more time to explain. I'm also not very familiar with that. I mean, I'm still understanding it. It's just last week's data. So I won't show you any. If I don't understand something very well, I shouldn't be presenting it. But I do know a little bit. The bottom line is, by this analysis, we are beginning to combine all of them together, not 2, but 3 components together. And we are beginning to find loci for some genes. You can also integrate this data with high C. So when you look at MEQTL, they're primarily in compartment A, which is the active compartment. Not surprising, but nice to show. You will predict that's how it should be, but should be is very different. Sometimes we take things for granted. Oh, what is new about this data? Oh, the new is we are showing it. We are not just talking about it. This is evidence-based study. You show evidence, MEQTLs, a lot of them in compartment A, EQTLs, EQTMs, compartment A. And you can look at different genes, how they're related, and I'm showing 2 examples, ABCA4, look at that, where they are going, and GSTP, another glutathione transferase, pi isoform. So you can combine and you can look at how the variants in these CPGs might be impacting what gene and how far downstream or upstream they might be. So we looked at that. Now I showed you MEQTL, and I told you about how methylation is important. But I didn't deal with aging yet. So we are beginning to do that. And thanks to, again, Dave Farrington, who has given us something like 250 retina samples, which go from age 20 to 107. And in each decade, except for 20, we have 20, 30, except for the first 20 decade and 30, where it's very hard to get 30, 40 samples. We have a little bit less there, but enough. But at every decade, we have 40, 50, 60 samples. So you can do now a lot of analysis. So this is a sort of a system that we have set up. You make DNA, you make RNA, we do genotyping, you do DNA methylation, you do RNA-seq, and we have done all that. And by doing all that, we are beginning to analyze the data in the same manner. After we submit the methylation paper, we are hoping to then focus on aging, and we will hope now to describe EQTLs and EQTMs and MEQTLs that are directly related to aging. And we don't know what that's going to happen. And this work, again, is being done by Milton English. He did the bench work, all of that, and Jayashree, who's doing all the study analysis. So this wonderful group of people now is beginning to take a look at how some of these things might be correlated to aging. And then we can do some sort of subtraction analysis. We have done the AMD part with AMD retinas, now with aging, and you can try to see how much aging contributes to AMD. Try to do that. We have a lot more samples to analyze still if you want to add for AMD actually, and we can then take it to macula as well as to RPE. Now just remember, these are more expensive studies, won't be possible probably at many places by a single R01. So thanks to NEI intramural program, where we can sort of utilize the resources to do this massive resource sort of organization analysis, which can hopefully be useful for a lot of people. So can we reverse it in some way? My friend Emily, beautiful studies that her group and with Tian and Keenan have been doing, and they showed that Mediterranean diet somehow is better. And it's sort of progression to AMD is slow. It also sort of allows you to maintain your cognitive function for longer time. You know, good epidemiology, good hypothesis, good sort of, we think it is working, it's nice. You look at clock, how will that happen? So you can say the age, chronological age is one, but biological age can be accelerated probably by smoking, sunlight, a bad diet, and maybe exercise, lifestyle changes, and supplements can maybe increase, rather slow down this progression to age deterioration. So we thought, can we test this hypothesis? So can healthy diet slow down the impact of aging? So we thought, all right, mice are not human. We can't do that in humans so easily because the variability is very, very high. We need to keep the genetics constant. So we thought, okay, you have a young epigenome. I mean, I'm considering 12-year-old, which is 40 years old mice or human as a young epigenome, but we can do 3 months. And then you can say, all right, now we can do experimental diet. We can have Mediterranean diet. We can have what we call American diet or high-fat diet, burger diet, and then AREDS2 supplements. We can have B vitamin supplements. We can do, so we did some of those. And then you can measure ERG, histology, mitochondria. You can do all sorts of things and see what happens. And then after 6 months or 12 months, you see what's going on. And Anupam is our bioinformatics person who actually has been leading this. And Daniel was a postback who worked on this. And we have done this study. We have some early data, but still being analyzed. We are looking at methylation. We are trying to see what can be done. But basically, all I'm trying to tell you is that we need a systems approach to understand AMD. We need to look at all components of AMD. We cannot really ignore aging. We cannot ignore environment. We cannot ignore genetics. They all are important and they talk to each other. You have DNA methylation. I put this in this slide in this review that Laura Campello wrote, who is now working on aging in the lab. There's a genome component which affects epigenome, and then there's a metabolic component, and there's a crosstalk. You can have metabolic, mitochondrial, oxidative stress and all, which can affect gene expression, chromatin and all that, and vice versa. They are in sync. They talk to each other. They don't exist in vacuum. They don't exist independently. They are together. And Mohita Gaur is another person who, another postdoc, new postdoc, who's looking at how metabolisms and metabolite affect epigenome, basically. And we are looking at that, and probably stay tuned for maybe at ARVO, some early studies we'll be presenting. So I already gave you all the conclusions, basically. GWAS signals are enriched in regulatory domains. AMD pathways operate as closely connected networks. Integration of GWAS and transcriptome has begun to yield some insights in targets. High resolution chromatin contacts with epigenetic marks can identify CREs and cis regulatory elements that are needed for retinal function. And super enhancers are associated with cell type specific genes. Genetic variants can affect expression of target genes by looping or by CREs. And I think our studies can be relevant much more than just AMD, but also glaucoma and other retinal diseases. And the more recent data with MEQTLs and EQTMs and retinal genome topology can allow us to link genetics to aging and environment. And basically, I think we need to have a much more coherent strategy to understand disease. And that disease could be any complex trait. Most common traits are complex and multifactorial. So I think I'm going to stop there. I already mentioned everybody's name. Just a tiny advertisement here. We have some postdoc positions. A bunch of these people are finishing and hopefully will be leaving with some faculty positions in the next year or two. So but our focus, the future is going to be towards spatial transcriptomics, spatial proteomics, epigenome, and super resolution imaging. If you're interested in that, do write to me over the next year or two. So I think I'm going to stop there.
Anand, for this wonderful talk, and I have to say this is just like my dream come true to talk about aging really as an important factor in those diseases. I invited people that have questions to tell me about it on chat. And then let me ask the first one. Did you think, or maybe you have some potential studies already lined up to correlate the GWAS or any other of your QTL studies with onset of disease?
No, that's a short answer. And the reason is that we really don't know at this stage where the disease begins and when does it begin. Our hope is, okay, I'll tell you how I'm thinking about it, at least. So much of this is just thought process. I believe aging is predisposing you to some of these other changes that are happening. You have these genetic variants right from the beginning, right from birth, but they don't lead to anything. So I think the aging study that we are doing is going to allow us to look at certain pathways that are really more susceptible to advanced age. And I think that is where the beginning of AMD might lie. And by combining that aging data with AMD GWAS or glaucoma GWAS or anything else, might help us sort of see what happens in 30s and 40s and 50s that eventually leads to these very bad phenotypes later on. I mean, I don't know if I answered correctly, but I just, it's just hard.
I know it requires also a lot of data to know the onset, right?
Yeah.
Yeah. So Zach, you have a question?
Hi, Dr. Swaroop. It was really, really nice hearing you talk. Really interesting work. So I have a question about kind of the onset of AMD and whether you have any instances of change in not only like the amount of certain transcripts that are expressed, but also the ratio of different spliceoforms. Maybe if you have like a longer splicing variant, crop up after the onset of disease or during the onset of disease.
So I'm sort of less interested in after the onset of disease, right? Because once the disease onset happens, there's so many things happening. We don't know what is cause and consequence. What we are interested is in early stages, right? What happens before. And that's why the aging study and the time course of early AMD. So we have samples from early AMD retinas, and we have from intermediate AMD. And so when you say early AMD atrophy and all that, so we have those samples as well with us. And we're trying to sort of now go down and see what changes might be happening. The trouble is we don't know whether they're related to age or AMD. So that's why this new aging study that we have will allow us to figure out what is age and what is AMD associated. So we are interested more in looking at 50s, 60s or early AMD, intermediate AMD kind of stage. and less on advanced AMD stage. So that data is sort of still a little bit early. I have to say one more thing which I didn't mention. Somebody may ask, and maybe they put, maybe they didn't think, there are many other tissues that are important for AMD, right? And we have only looked at peripheral retina. And the reason we're looking at peripheral retina is because Emily and others will tell you that AMD just doesn't start only in macula. You have peripheral components as well. But not only that, but it was easier to do, right? You take the low-hanging fruit first. Now we have done with macula, and we are finding new loci and new things that are coming up, new information that did not show up in peripheral retina. And we are hoping that we'll have enough samples from RPE. And when we do that, maybe that will fill a lot more gaps. Maybe that was the thing that we may need to do. And then maybe with endothelial cells, same thing is true for glaucoma. So I think each of these fills part of the puzzle. This is not, retina also data allows you to look at many other things just beyond AMD. So that's why we focus initially on that. Yeah. But what you are saying is, again, our focus is on this early intermediate AMD kind of stage. But we are still not able to dissect what is aging and what is AMD until we get some new data.
Thank you. I'd like to choose Dr. Zach for the last comments. I know that he's also interested in the aging in both, in all retina. And then before now, maybe Dr. Kefalov would.
Yeah, thank you. That was really beautiful talk. My question is, obviously, this is a very powerful approach using the kind of system analysis, looking at genetics and epigenetics to understand better complex diseases like AMD. So I would love to hear your thoughts on how you think that eventually informs the development of therapies. Do you think that you'll be able to come back again and narrow it down to a few genes that are really important and kind of remotely control everything? Or do you think that the image will be even more complex and even more difficult?
No, I'm a chronic optimist. So my idea is not to treat the disease after the symptoms have come up, but sort of tackle it before it really happens. So that's why I keep coming back to this 40s, 50s, 60s type of age phenomena. If we can understand what is going on over at that stage, and by whatever means, if we can slow down the aging by diet or vitamins, or we can sort of slow down by targeting a pathway, one specific pathway, maybe IGF pathway, or maybe something, maybe TGF beta, or maybe some inflammatory response. I don't know that as yet, but what I'm saying is, in that early stage, I am really hoping that we will have one or two pathways that will stick out, that are really critical sort of at that stage, which may make it worse. But we won't know the whole story just from retina data. So that's why retina, macula, RPE, and maybe endothelial choroidal cells. I think when we have all of that, then we will have. But I think we have set the groundwork. The analysis and everything else should be much more easier. The first time it takes 3 years to do it. Next time it takes 1 year. Third time probably 4 months or something. So I'm just hoping that once we have the pipelines and everything set up, this should go much faster and will allow us to look at some of those very targeted pathways which can be used for therapy. So
early diagnosis also.
Yeah, early diagnosis to everything else. So yeah. Based on genetics, based on phenotyping, I mean, clinicians have wonderful insights into, how to look at some of these things. So a combination of these things, I think, is going to be help us, and hopefully in my own lifetime, not too far away, in the next few years before I retire, if I do.
So we have still several questions here. Cesare? And if I can ask you to repeat the question, some of the
okay, sure. Okay, absolutely.
Thank you.
Fantastic. So I have a question. Do you think that the analysis of genome and epigenome with lowering costs, so the costs of the analysis are lowering, do you think that it could be potentially employed as an early intervention in younger individual by disease suspects to maybe not design, but offer some kind of information for the life ahead to reduce the risk of
development of certain conditions.
Yeah, so the question is by Tejare whether we can use the new technologies that are becoming easily available with low cost. Can that information be used for future either prevention or therapies? I would add the word prevention as well. And I sure hope so. I mean, I heard Francis Collins, I think, or someone, not my idea, to NHGRI folks, that maybe in a few years we can have some chip under our skin or something, which will have all our genome sequence. You go to the doctor and they scan you and say, okay, you have these bad genes or bad variants. Maybe this is what you should focus on. So sure, theoretically you could do that. And same thing can be true for retina, even though it's not easy to get biopsy sample. But if with new technology, all you need is a single cell or few cells, theoretically it might be possible to take a little bit of vitreous, a little bit of something. I don't know, I'm just, again, guessing here, to get few cells to look at your epigenome even, and in those cells, and see how things are progressing, and maybe follow up and try to advise you what to do and what not to do. And again, I mean, if there are some therapies available. So genetics can be very, very powerful tool for diagnosis, which can help the clinician. At this stage, clinicians probably have better idea as to how to diagnose the disease. Genetics still is not as advanced, but we're hoping that with more information, I think in the next 10, 20 years, probably it will be and can help.
And now we have Sam.
Hi, Dr. Swaroop. A really great talk. How does the circadian rhythm affect the topology and the organization of the genome?
You know, so how does circadian rhythm affects topology of the genome? I think it's a great question. We have thought about it, to tell you the truth, in mice. I don't think we can do that in humans so easily, but in mice we are doing some, we have done the early parts of that, gene expression to methylation and other things, but we haven't really gone into high C. Again, as I was showing you, some of this work is fairly new and it took us a long time to understand. And we are now going to go to mouse part with some of these. And again, there are different problems with mice. With doing some similar analysis at high resolution. I think the gross changes in genome topology will not be there. That's my guess. There may be some very specific targeted changes that may be happening. I think circadian is more of a signaling, going to be a signaling problem rather than at the genome level. I think the genome level changes are going to be more related to long-term or chronic diseases, or or aging diseases, or something. I mean, that's my. I think if cell has to do something very quickly, I think changing genome topology doesn't make much sense. I might be wrong. Okay.
And I think we have last question from John, and then we will hear the comments.
Dr. Swaroop, great presentation. So have you ever, as you explained early on, a single individual can have multiple variants present. Have you ever looked at not just a single variant association, but like the existence of multiple variants at the same time, leading to perhaps resilience to development, or association to resilience, or association to susceptibility, and perhaps that informing pathways?
Yeah, so John is asking, right? So he's asking whether we or someone has looked at multiple variants at the same time in single individuals, and whether they can sort of either enhance your probability of disease or decrease the probability of disease. I think that is a research that is ongoing right now. We did some analysis in the 2014 review. We were very curious to see how the role of complement. We were able to explain about 50% of disease with complement, but not area based on area under the curve. If there were individuals who had few individuals who had all complement or all means many complement susceptibility alleles at that time that were known, but still didn't get the disease. And then there were others who with less, but no, still had the disease. So just one pathway or one set of variants is not sufficient to get you disease. You have other things that, and that's why we thought of threshold, that you have to really reach a certain stage, and that stage can be reached not just by genetics, but combined genetics with age. Again, remember, these variants may be present even at birth. They are present even at birth, not they may be, they are present at birth, but still we don't get disease. So there's something more is happening to it. And that is very variable. Everybody ages differently, environments are different, lifestyles are different. So what happens there is still unknown. So genetics is very important. Don't get me wrong. I'm being a geneticist, I would not say that this is not critical, but they predispose you. So we have looked at it, but yeah, there is high probability. Your probability goes up. If you have all the bad genes, you have threshold already reaching here, you have very little to cover, right? So to go over the hill. So yeah. But if you have everything is, maybe 10 different genes are all, so yeah, maybe you get, but then why wait for age then, right? But you still, age is still there.
So now, Don Zach, Dr. Swaroop is all yours.
Well, thank you. It's a great honor. Anand, as you know, having been friends and colleagues for more decades than I want to remember, this was really an amazing tour de force of how you've integrated so many high throughput technologies to concentrate. So really congratulations on a beautiful study. And I think as you commented, this is really a nice illustration also of what the NEI intramural program could do, because it'd be pretty hard for most independent grant funded administrators. So, sometimes the NEI reviews always ask, what are you doing that takes advantage of NEI that you couldn't do at your average great university? And I think you've really beautifully demonstrated what can be done. Great model. So I just wanted to ask you two related and probably unanswerable questions. But anyway, one is, given all the hundreds of retinas for which you have data, do you have some personal information, history? Is there any information about the donors of these eyes that could be integrated into your studies to add, just one more dimension of your correlations? And then part two of this is, I don't know if you have blood samples from some of these samples, and again, I realize it's a big endeavor to do that, but if you did similar studies on the blood versus the retina, even though obviously lymphocytes are very different, could you use machine learning or some kind of approach to pick up a way to predict from blood samples something about the retina, even though it's different in the blood? Does it provide insight to what's going on in the retina?
I think both great questions, Don. Yes, we have been friends and collaborators, colleagues for decades now. So let's not go back how many, some of these kids were not being born at that time.
Back when we both had more hair, I guess, yeah.
I don't know about you, I had a little bit more hair. But yeah, so to answer the first question, yes, we do have some data. We talked to Deb about those samples and data. So we have smoking status in some cases. The trouble is when you incorporate smoking, the number of samples, the power sort of goes down a little bit. We have now gotten more samples from her, and we are planning to analyze that further, incorporating just the smoking data set. The other data, the medical data, was not as elaborate, again, for everyone, and that creates problem. But I think we can incorporate smoking into it. So that's, but I think that's a great point. Maybe we need to talk to Deb some more and see if we can get enough power. The second question, I think, is something that obviously will be amazing to do. We don't have any blood samples from those, and I don't think Deb has blood samples from those, but I should check. I think our thought was slightly different. We thought that once we find some interesting pathways or whatever that we may think may lead to blood metabolite or some blood sort of markers, then Emily has got thousands of ARED samples, serum from many of these. I think that could be one way then to look at, but we at this stage don't have any good idea before we thaw some of those samples and waste that precious resource. So I think that essentially we are thinking of doing, but maybe in a slightly different way than what you were saying. But I think I should talk to Deb, and maybe she has. I don't know. We never asked. Yeah.
Thank you again. And we have one more thing here.
Dr. Swaroop, on behalf of the Department of Ophthalmology, the Center for Translational Vision Research, I want to thank you for your visit and for this very insightful and thought-provoking talk, and present you with this distinguished speaker commemorative plaque.
Thank you very
Thank you.
much. Thank you. Thanks. Thank you, guys. Thank you for listening.